acids, remember, they spell protein, could order themselves into meaningful biological sequences on their own without pre-existing genetic material, being present just reached, I guess for me, the intellectual breaking point sometime near the end of the 70s. So less than 10 years later, he realized this book he wrote was a futile effort. There's just too much obvious and inherent and rational observation of a design to try to pretend there's not a creating architect behind this. I appreciate Dr. Kenyon's honesty. What he was talking about, the fact that there's no genetic material present. In other words, you need DNA to make proteins. Let me show you the cycle. DNA is involved in the manufacture of proteins. And he's saying there's no way these amino acids could come together in a meaningful sequence without the DNA and all of its storage. Remember, each cell has more information stored in that DNA than all of the Encyclopedia Britannica. Okay? And so what he's saying is there's no way these amino acids could spell words. It's like putting a chimpanzee down in front of a typewriter and saying, "Go at it, boy." And eventually, he's going to write a Shakespearean novel. Now, he may accidentally hit "the," and he may accidentally get a word. He may even pull off a sentence, maybe even a paragraph. But he's not going to write Hamlet with all of the entrances and exits, no matter how much time there is, because each time that chimpanzee's hitting that typewriter, he's starting over. And so he's saying there's no way these amino acids could accidentally put themselves into meaningful sequences that start self-replicating without the presence of DNA. But here's the real trick. Here's the real rub. The real positive evidence for design. DNA is needed to make all these proteins in the cell. But guess what's needed in the manufacture of DNA? The very proteins it makes. Now, do you understand the implication of this? DNA manufactures the very proteins that are necessary for its own existence. They would have had to both randomly involved, accidentally in the primordial soup at exactly the same time, and at temperatures in the primordial soup that are destructive to DNA material. When we do experiments with proteins and DNA and stuff, we extract proteins out of rat liver or whatever, we store them at minus 70 degrees to keep them from breaking down. These things don't happen at minus 70 degrees. So even if these things spontaneously came together, they would be destroyed instantly. So what we have here, we have thousands of proteins in a single cell, from 50 to thousands of amino acids making up just one protein word, spelling a word. DNA, in this process we're about to witness, is what manufactures those proteins. And some of the proteins it manufactures are essential to it self-replicating into further cells. Now, we are getting ready to look at what's called the transcription process. We're going to go inside a cell and then go inside the cell nucleus. That's kind of like the little brain of the cell. That's where this genetic material is created. And some of this genetic material breaks off and it becomes a messenger and it leaves the nucleus of the cell, and we're going to watch a protein actually being built. Then we're going to watch a protein being folded. And then we're going to see as this protein is carried off by another molecular machine to the place in the city of the cell where it's going to do its work. This is an astounding discovery that we have made of the amazing factory work within the cell and how DNA is essential to proteins and proteins are essential to DNA. And it's undeniable. And then the man who speaks at the end of this clip is Dr. Dean Kenyon.